Welcome back, everyone. Today we're going to be going over chapter 10, which is dealing with genetic engineering. Here we're going to be applying a lot of the concepts that we learned about how DNA essentially works from the previous chapter and using it to understand how we've essentially hijacked that to work for our benefit in science and medicine. Okay, so our objectives in this chapter, we're going to define what recombinant DNA even means and what a genetically modified organism is. We're going to use appropriate terminology, of course, more terms. Of course, we wouldn't want to leave those out, and how we would refer to the sizes and lengths of DNA, which is something that you might get tested on at some point. How do we create recombinant DNA? What are some of the products of that process that we've been able to, I would say enjoy or benefit from in modern society? What is PCR and how does it work and what can we use it for today? And what is gene therapy and how would we use it to cure diseases? What's the concept behind that? How would it be useful? Okay, so getting into this, there's two basic kinds of science. I can always forget that my bracelet triggers this thing. There's two basic kinds of science. There is like basic science, which talks about here where in basic science, there's no product or application. You're not trying to make something to make money off of. You're just trying to understand it for the sake of knowledge. Okay, that's all basic science is. It's just for knowledge purposes. But then we have applied science, and that is essentially using information from basic science, that knowledge that we've learned, and using it to gain something from it. So now we have, learned, okay, this is how DNA replicates. And now we're going to use that information to say, okay, we know how it replicates. So how can we use that for our benefit? Let's design this thing called PCR or whatever. It's more complicated than that. I'm sure you're aware, but that's the idea behind the difference between applied science and basic science. If you were asked to, describe the differences between the two, applied is just using the knowledge from basic science for a gain, for a use, applying it, right? Applying the knowledge from basic science for a use, for a purpose, okay? Applying it, let's describe it. I hope that makes sense. All right. That's not what I meant to do. Okay. So you guys remember DNA, our good old friend DNA, our double strand here. When you have DNA and you have your double strands right here, if you heat it, there's some chemicals that you can use to do this as well. But the most important thing that I want you guys to be aware of for this chapter probably is heating. When you heat the double stranded DNA, it separates into separate strands. That's very, very important concept for this chapter, okay? When you cool those strands back down, they're going to come back together again and with their appropriate base pairs matching up just like they did before. So they can come apart, and then come back together based on temperature, and we can control that using temperature. So that's the concept with that information. Additionally, okay, so that's how DNA can behave overall. If you want to get specific and you want to cut DNA at specific parts or, separate it at a specific part, and let's say you want to insert a new piece of DNA into something else, maybe my genome, you don't like my hair color, so you're going to give me the genes just so that I can have, red hair or whatever. The way that you're going to achieve this doesn't have to be, but can be through using these things called restriction endonucleases. And we're going to talk about all of this stuff soon. So what is a restriction endonuclease? A restriction endonuclease, essentially we're going to clip DNA crosswise at selected positions. What does that mean? Literally, like we had our DNA, our double-stranded DNA. There's some space here. Our double-stranded DNA, and there are specific places where these enzymes, remember enzymes are proteins that have functions. These enzymes are going to come in and recognize specific sequences in that DNA and cut it at those specific places. Very specific, always going to be the same place. You can predict it. So then if you know that these enzymes are going to cut at that spot, you can design your DNA piece that it'll come and match in with that spot. You can insert it into that little spot perfectly. I have some pictures to show you what I mean by all that, of course. But okay, so the enzymes themselves are called restriction endonucleases. I know that's a big, long word, but that's what they're called, man. I don't know what to tell you. The restriction endonucleases, we believe they were enzymes that bacteria used or actually still use to defend against foreign DNA that they didn't want around. So they could like cut it up and it wouldn't work. So that's what we believe that they were originally used for by bacteria, but now we utilize those enzymes for our purpose in genetic engineering. Okay. Predictable spots that they'll cut at. Okay. So like, oh yes, and I do want to be specific here when I say it protects from foreign DNA, bacteria and archaea. Bacteriophages, which are viruses that infect bacteria, right? And plasmids, little bits of extra circular bits of DNA that carry genes that can make the bacteria more virulent or might not be helpful for the bacteria at all, but they can't really control that. They don't pick and choose that. So if they don't want it, they might want to cut it up, right? Get rid of that trash. So that's what restriction endonucleases did naturally in nature. What do they do for us now? So endonucleases will recognize a sequence that says 4 to 10 base pairs, 4 to 10 base pairs here. And they clip at what we call palindromes. I'm going to show you what I mean by that. It's the same forwards as it is backwards, like race car. Okay. Literally the word, right? It's the same forwards and backwards. And then it creates a sticky end, which means they say staggered symmetrical cuts. They are like, it will like, if for your double strand that looks like this, that will get cut by the thing. If it reads this here, then you might have it look like this. And then the other side will look like this. So you see how there's like what this extra overhang that's going on there. Those are the sticky ends. That's the idea of the sticky end. Why is that useful? Because that helps whenever I come in with my extra DNA. Hey, I've got my crap that's also been treated with the same stuff. And it's going to have this overhang here. And then this end's going to have this overhang here. And it can match up with its sticky ends onto these sticky ends, the way that we've designed it to. And they'll match up perfectly. That's the usefulness of the sticky end, just FYI. So it's not a perfect, it's not like it cuts the double-stranded DNA. It's not like it cuts it into perfect little parts that are perfect and blunt cuts at the end. There are some that do that, but I'm not worried about you guys knowing that. I want you guys to be aware of the sticky ends because that's the useful part for us to understand how the recombinant DNA is going to be prepared. Okay. All right. So here's some pictures about it. You know, here are some of the most common ones. These Eco R1, probably Eco R1 and Hind 3. I mean, I don't know. These are some of the most common ones. Okay. So just if those saw them written out on a test or something, you would be able to recognize them as some of the most common restriction endonucleases. You can't say I didn't show it to you. So you see how this is GAATTC, and you see how this one in this direction, this one goes, the top one in left to right, and the bottom one right to left, right? Because 5' to 3', and then 5' to 3' the other way. All right, so we got that lined up. They're the same. GAATTC is the same GAATTC in the other direction. That's a palindrome like that. The other way you can look at it is, look at this. You have GAA. What does A bind with? T, right? It shows us here, right? It's not a trick question. T, T, C, right? For the G, binds with a C. Well, look at that. It's the same. So that's the idea with what they're talking about with the palindromes and how it cuts stuff. Can you not move around so much, Ripley? Please don't move around. You chose to sit there. Commit. Okay, so same with this guy over here. If you were to look at that and break it down, that you could say the same crap about it, right? So palindromes, and they're going to cut. So let me get rid of this crap that I put on here so that you can see what I mean by sticky end. So that's the sequence that it will recognize as far as the palindrome. How does it cut? The actual enzymes will cut and create cleavage points through these lines that we have drawn on here, okay? And that will leave those sticky ends. And this is a depiction of what will be going on in those sticky ends, okay? So we have our site where we're gonna cut the DNA. This is our double-stranded DNA over here. We're recognizing the sequence. We've created the cut. We have a sticky end, a C tag, and a GATC. And those would match up with one another, right? Got our sticky ends. So this is for the original genome, the original chromosome, or the original DNA. Okay. Now we're going to put some DNA from something else in it. From, I don't know, me. This is my brown hair gene or whatever. We're going to put it in some bacteria. I don't know. It doesn't matter, right? So we have a gene that we've cloned from something. We know it's sequenced. We've made it out synthetically, and we have created it so that when we treat it with the same restriction endonucleases, it creates the same sticky ends, so that when we mix a high concentration of what we're putting in with the appropriate concentration, a lower concentration of the vector of what we're putting it into, right? What's going to carry it? Those sticky ends will stick together. They'll match because they're complementary base pairs. We've set that up that way. So then, we can, cool it down a little bit, help it stick, and we'll treat it with DNA ligase, which, smooths over all those not quite linked together nucleotides that aren't quite stuck together, just like the Okazaki fragments during all of that replication. So yes, that's what's going on there. So now we have created whatever the original DNA is with the purple. And we've inserted, Lauren DNA into it, and it's seamless, and it can read it just fine, and it works, and it's happy. It's the correct, orientation. 5' and 3' are matching up, and all of that. So that is how we have used these as tools to insert genes that code for specific things into vectors. So it could be plasmids, and we can put those plasmids with extra bits of DNA, right, that bacteria can take up. We can put genes into plasmids and force bacteria to take those plasmids up into themselves and then express whatever those genes are that we put in the plasmid. And we can make that triggered as well. So that's the idea of how all that works. So a restriction fragment. So we got all this, we're cutting things up and whatever. A restriction fragment is a piece of DNA that we produce by cutting with restriction endonucleases. Restriction endonuclease, you cut it, you get restriction fragments. Pretty straightforward. Then we have this thing called a restriction fragment length polymorphism. That is essentially a difference in the cutting pattern of endonucleases can give rise to restriction fragments of different lengths. And that will allow for comparison of DNA of two organisms. What does that mean? My genes are different than your genes. If I treat my whole genome with EcoR1 and your whole genome with EcoR1, how EcoR1 has cut up each of our genomes will be different from one to another, or especially from like my genome to Ripley, my dog's genome. So that's what we're talking about there. Restriction fragment length polymorphism. Polymorphism is dealing with differences in the things. We can use that for identification in forensics. Okay, so that's how that would work. Let's go ahead and watch. Hopefully this will be loud enough for you guys. My dog is snoring again. She's really old, guys. Go away.
Restriction endonucleases are enzymes that cleave DNA at specific nucleotide sequences. The sequence recognized is often 4 to 6 nucleotides long. For example, the restriction endonuclease EcoR1 recognizes the sequence AATTC. The nucleotides at one end of the recognition sequence are often complementary to those at the other end. The two strands of the DNA duplex have the same nucleotide sequence running in opposite directions for the length of the recognition sequence. Because the same recognition sequence occurs in both strands of the DNA duplex, the restriction endonuclease can bind to and cleave both strands of the DNA molecule. Because the bond cleaved is typically not positioned in the center of the recognition sequence, and the DNA strands are antiparallel, the cut sites are offset from each other. After cleavage, each DNA fragment has a single-stranded end that is a few nucleotides long. The single-stranded ends of the two fragments are complementary to each other. These single-stranded ends, sticky ends, can pair with each other. Once their ends have paired, the two fragments can be joined together by the enzyme DNA ligase, which reforms the phosphodiester bonds of DNA. What makes restriction endonucleases so valuable is that any two fragments of DNA produced by the same restriction endonuclease can be joined together. Restriction endonucleases are fundamental tools in genetic engineering.
Okay. Okay, ligase, getting back into this. So we have cut our things with the restriction endonucleases. We've made our restriction fragments because of that. We're able to stick our, target gene into the vector of whatever we're putting it into. Now we need to make sure we seal it up, tape it back together, rejoin those phosphodiester bonds, as the video was saying. Just important that you guys understand that ligase is just going to help rejoin and seal everything back together. Okay, so that's going to be ligase, DNA ligase. It's going to seal it all back together. If you don't seal it back together, it doesn't work. It's just like pointless to have even done it. Okay, so what enzyme do we use to glue the target gene into a cloning vector? And that's just what we were talking about just now. That is that DNA ligase that's down here. Okay, right. So you guys remember about how eukaryotic genes have these things in them called introns and exons, and that in order to go from DNA to, so if we're going from DNA to mRNA, that messenger that carries the gene information, in order to create a protein from that for the ribosomes to read it and create a successful, protein from that, we have to cut out these things called introns. Okay, the thing that we use, the term, not the thing, I should be clear about this. The term that we use to describe the removal of the introns is something called splicing. Literally, splicing just means you cut out a part of a DNA or RNA, in this case, nucleic acid, and you've rejoined it back together. So we just cut out that intron. Okay, it will look like kind of like this. This loop part is the part that you don't want that we're cutting out, and then the ends of what we do want come together. So that's the splicing process, right? So that gives us the mRNA that we can actually use to make a protein. If we put eukaryotic, like our cell DNA, into a bacteria, they can't deal with that. They can't deal with the introns. It's impossible. So we have to almost reverse engineer our crap that we're putting into the bacteria. So what we have to do is take the useful mRNA that actually gives us a product and create DNA from that using the enzyme called reverse transcriptase, making DNA from the mRNA. Then that cloned DNA or cDNA, we can use that cloned DNA to put into our bacteria or whatever. So if we're trying to make insulin, have bacteria make human insulin, we have to take the introns out of it. And this is how we would get to that. Okay, so it's important to note this is a thing that happens with eukaryotic DNA. So again, to reiterate, eukaryotic genes can't be cloned from the chromosome directly and expressed in E. coli, which is a bacteria, because E. coli is prokaryote. Eukaryotes have introns that have to be clipped out in order to make the product successfully. Okay. This is just showing us gel electrophoresis of DNA products. You use the enzymes, whatever enzymes, could just be one, even if it was just EcoR1, it might cut things in a number of places. You cut it up, you prepare your, you look at the samples in the gel and run them out on the gel, and they will separate based on size. Now, if we're running things on gel electrophoresis, the size is going to get, the larger things are going to be closer to where you started, the wells that you put the sample in originally in the gel. You put them in the little well, you literally inject them with the little pipettes. You guys have seen it, right? And then you put electricity through it. Remember, DNA has a negative charge to it. So the negative DNA is going to move towards the positive electrode. Okay. DNA migrates towards the positive electrode because DNA is negative. The larger molecules on top of that, the larger molecules are going to be kind of stuck in that gel. They don't move as easily in the gel or as quickly in the gel. So the larger ones are going to be at the top where you started. The smaller ones are going to move faster through it. Okay. They can sneak through, like if you, I don't know, had a really, tiny guy who was nose guard on the football team or something like that, right? Sneaking on through. So yeah, DNA is going to migrate towards the positive, and we're going to start with larger. It's going to get caught up towards the wells, and the smaller is going to move faster towards the bottom. Okay. So what would increase the speed that DNA would migrate in one of those gels? It's called an agarose gel. So how would we increase the speed? You can, if you shorten the length of the fragment, if you decrease the charge of the fragment, well, no, the charge, the more charged it is, if it's more negative, it should move technically faster. If we decrease the charge gradient, again, that's not going to help it go faster. Increasing the amount of agar in the gel is just going to catch things up more. So that would be your A option, shortening the length of the fragment so it can move faster through the gel. Okay. Moving on to nucleic acid hybridization. Here, we're not doing any electrophoresis in order to compare sizes here. We know whatever the sequence is for this particular gene that we're interested in, okay? We just want to find which bacterial colony has the plasmid or whatever that we wanted, the target thing that we put into it. So we've created a gene, we put it into a plasmid, which bacterial colony on our plates actually have that gene in there. And you want to know, maybe make it based on a mixture or something like that, or which bacteria just naturally have this particular gene or whatever it is. It doesn't matter. What you'll do is you'll grow your bacteria. You put a filter paper on it, just a piece of filter paper. It's just paper. And then you'll wash it a little bit. You got to do that because that you don't want other stuff interfering. Next, we have it washed, and we have, we're breaking open our cells. We're going to treat it with sodium hydroxide. Why? That's going to separate the DNA into single strands. We said we could do that with heat, but I did mention we can do that with chemicals. This is one of those chemicals. Sodium hydroxide will force those double strands apart into single strands. So we'll treat it with that to separate it out. So we've got our bacteria. Now we can treat with a specific probe for whatever the friggin' gene is that you're interested in. And if it binds to the thing and hybridizes and just like matches up like what we've been talking about this whole freaking time, guys, it's not that hard to understand. We separated the strands, put something on there that should match with your specific gene sequence. If it is able to bind to it, it'll bind to it and it'll stick. And we'll wash it and stuff and make sure there's no background going on. But if it sticks, then those colonies are the ones that you would want to know about that are carrying your gene of interest. Okay. Right. So this is what's talking about gene of interest versus the DNA probe and whatever. So now we have the film that shows us that these colonies here match up. And those are the colonies that contain whatever gene you're interested in. And, Right. So it's talking about making a replica master plate, and that's kind of a little harder to understand. I don't need you guys to know about it too much, but like you would literally have taken like another filter and put it onto another plate so that it would match up literally exactly like this colony matches this. So you could grow it up and then have those bacteria available again. So now you can move on with whatever research you're doing. But anyways, that's the concept behind this. Using probes to match them up to the exact sequence of whatever gene you're interested in, and doing some rinses and whatever. And then if it matches up, then that what colony has your thing that you're interested in. Good. This is a video of it. I hope that it explains it a little bit more. Because I feel like videos on this stuff a little bit more useful. Let me put the captions on since apparently it's really quiet and I have it turned up all the way.
At high temperatures, double-stranded DNA will denature or separate into single strands. When the temperature is lowered, the two strands will anneal because of the base pairing interactions of the complementary strands. If the nucleotide sequences are similar, complementary strands from different sources will also anneal. This is called hybridization, to indicate that each strand of DNA came from a different source. Regions of double-stranded DNA in which the respective single strands hybridize to one another are said to be homologous, or have similar or identical nucleotide sequences. The high specificity of base pairing interactions between complementary strands of DNA can be used to locate a specific nucleotide sequence in a sample. If the DNA from one source is immobilized by attachment to a solid surface, such as nitrocellulose, homologous DNA from another source will hybridize and be retained by the immobilized DNA. This is the basis for various DNA probe techniques. Non-homologous DNA will not attach.
So non-homologous, meaning like it doesn't have the sequence that matches. I know this is still pretty new stuff for you guys. Okay, so let's imagine that we were trying to do this and we would want to know which of these genetic probes would hybridize to this target sequence given to us here. And I want to be clear, this is part of one of those things where you have to be aware of when you get these questions. The fact that you have to think about things should be 5' to 3'. So if I write out the matching thing to this would be C, T, A, G, G, T, bear with me, A, T, A, G. Okay. This, however, would then have to be 3' to 5', because that's how the complementary strand is. So the answer that you would be looking for would be read in this direction because all of our choices start with 5'. So we are looking for GATAT, whatever. That's this one down here. Agreed? I hope so. Hope you guys agree with me. Okay. Sizes of DNA. We're talking about them in terms of their base pairs, literally how many base pairs they have, G, A, T, C, whatever. Literally how many of them. So palindromic sequences, remember, recognized by the endonucleases, we saw ones that I think were like 6 to 8 or something like that. But anyways, they can be 4 to 10 base pairs. Okay. Literally like 4 being G, A, T, C. Okay, so average E. coli gene in E. coli, their average genes are about 1,300 base pairs or 1.3 kilo bases. Kilo being 1,000. So you can break it just like kilometers and meters. Okay, so the entire genome of E. coli, we would say, is about 4.7 million base pairs or 4,700 kilo bases. It's another way to break it down, or 4.7 mega bases. That's million. These are the terms that you should know. What base BP, what is that? KB or KBP, that's another way we might write it, because we don't always just put KB. KBP is pretty common as well. And then mega bases or MB or MBP. Okay, so those are the terms you need to be familiar with as far as the sizes go. If I were to say which one is bigger, and I were to give you these choices, which one would you choose? A or B? You should choose B. Why? Because 10 kilobase pairs is 10,000 BP. Okay, so 1,000 is not as many as 10,000. So when you see the K, think 1,000. It really is just like what we see in our everyday life with K. 10K, 1,000. Don't get tripped up by the fact that when you guys are looking at these on the test, because these are questions that you're going to see, I guarantee it. If not on the lecture exam, you're going to see it on the lab exam. And it's going to appear, I want to say more than once. Okay. When you see 10 or you see 1,000, stop and look to 1,000 what? Base pairs or kilobase pairs or megabase pairs? Like, what are we talking about exactly? So take a step back, take a breath. What exactly are we talking about for each choice, please? Okay. I don't know why that always trips people up. You think it seems simple right now. I promise you won't get it. I'll say you. I'm sure you're smarter than the others. Anyways, you guys get it, right? So don't let that trip you up when you're taking the test. And it won't be simple thing like 1,000 versus 10. It'll be a little more complicated. So moving on, Epstein-Barr virus has a whole genome of 172 kilobase pairs, whereas the human genome has 3.1 billion base pairs, which terabases is a billion. Like computers, yes. Stop moving, dude. All right, moving on to the PCR. PCR, the polymerase chain reaction, the molecular Xerox machine, one of the most useful tools in biology. This will rapidly increase the amount of DNA in a sample without the need for making cultures or carrying out complex purification techniques. Take a very, very small, a very minute amount of DNA and amplify it to millions within just a matter of hours. So that's what PCR does. It just makes copies of an original to make it enough of it that we can actually use it for a thing. That can be identifying what the thing is, that like what DNA is this from? We can use it for that, or use it for like cloning and stuff later on. Okay. All right. So that's the idea. So it's pretty sensitive. It can detect cancer from one single cell. It can give you one cell, pop it into a PCR machine. I can tell you whether or not it's cancer, has cancerous genes in it. And then it can diagnose an infection from a single gene copy. So that's going to be genes that are coming, that are coming from the like virus itself. Okay, so it's the same thing as DNA replication. Remember, this was DNA replication over here on the right. We open up the double helix. In this case, we're using heat to do that. It's not using, topoisomerases and all of this. We are going to open up the helix. We're going to expose the strands as templates. We're going to add primers. Those primers are going to serve as things that the polymerase can build off of. And then we're going to have the action of the DNA polymerase itself. Shush! Be quiet. Stop. Stop that. Okay. So we do have to have primers here. That's an important ingredient. So here are, yes, do be aware of these ingredients, right? If I were to ask you which ones have to go into it. Number one, primers. They are, we're going to make those. Just, we're going to be quiet. We're going to make these primers to match up for a specific gene, right? So we add those into the mixture. So those are available for our polymerase to build off from. Remember, it can't just build off from nothing. It has to have something to build onto. This shows it where to start. Okay, so that's what the primers are. They're usually 15 to 30 bases. And again, just it'll be specific to specific target sites. Tells the polymerase where to start. Okay, based on whatever your gene is. Number 2, we have to add a DNA polymerase. We have to have a special polymerase for this because of how this reaction is going to work that is resistant to high temperatures. Okay. So we have a special polymerase that we got from thermophilic bacteria, the ones that are, good with high temperatures. It's called Thermus aquaticus. That's the bacteria. So we call it TAC polymerase. T from thermus, AQ from, aquaticus. So TAC polymerase. It's resistant to that heat. So remember, a lot of proteins can get broken up by the heat. We're using heat to split up the DNA. So this has to be a polymerase that can withstand that heat, right? Has to work in the heat. So that's why we need that special DNA or that polymerase. All right, the next thing that we need, whatever we're trying to amplify, that's important. Whatever the thing is that we're trying to look at, right? And then the buffer. Why is the buffer important? It's not just for keeping everything like in a liquid. It's also to provide a source of the nucleotides. That's what we're adding on, right? The A's, the G's, the T's, those have to get added on as we're working through with the polymerase. So that's the source of those nucleotides. And it provides things that the enzyme needs to work, like magnesium. This is a picture of how it works. It is very straightforward. And it might not look like it in the picture, but it really is, I promise. You have a double-stranded DNA. You're going to heat it up to separate it. Then you're going to add your primers. They're going to stick at their whatever specific points, okay, on that split up DNA. Now that we've had added the primers on, the DNA polymerase can add the complementary part to whatever its strand was. So now we have gone from one single strand up here to two. Well, I say one single strand, we have a double-stranded target sequence, but one source. And now we've got 2 copies. Then we're going to keep doing that over and over and over, and you're going to double it with each step, right? So it's exponential. So we're going to go from 1 to 2, 2 to 4 copies, 4 to 8 copies, 8 to 16, 16 to 32, and so on and so forth. And this happens very quickly. And if you do this for hours, you are going to have millions of copies at the end, exact copies of what you started with. That can be used for cloning, or we can use it to determine what the thing is that you're looking at. Like, we can run these reactions and read them in real time if we have building up and making of copies of something, when we only provided it with primers that would allow for amplification for salmonella, and we see amplification, it is positive for salmonella. And you can say that in real time. So that's a useful thing for diagnostic tools, right? And it's very, very specific, and you're not going to get like very many false positives at all. Like, it should be very, very specific. If it's positive, it's positive. So that's the beauty of all of this. Okay. So this is going to talk to us about how exactly this stuff works. Let's put this on first.
The polymerase chain reaction is a method for making many copies of a specific segment of DNA, starting with a very small amount. This technique can be used to identify specific microorganisms from a small amount of DNA, and to identify persons involved in crimes from DNA on cigarettes or in a single hair follicle. The DNA to be amplified is mixed with deoxyribonucleotides, a thermal stable DNA polymerase called TAC polymerase, and DNA primers. The DNA primers hybridize to the ends of the gene to be amplified and provide a starting point for the TAC polymerase. The mixture is heated to break the hydrogen bonds in the DNA, forming single-stranded molecules. The mixture is then cooled sufficiently to allow the DNA primers to anneal to each end of the segment to be copied. TAC polymerase then synthesizes the complementary strand of DNA using the primer as the starting point. The temperature is raised again to separate the DNA strands and then lowered sufficiently to allow the primers to attach. TAC polymerase now synthesizes another set of new complementary strands. This process is repeated until enough DNA has been produced to be identified or used for further research. After 21 cycles, one molecule of DNA can be amplified to over 1 million copies. This amount of amplification can be achieved by running the reaction overnight in a thermal cycler, an instrument that automatically raises and lowers the temperature at appropriate time intervals.
All right, so that's the concept behind polymerase chain reaction, PCR. So when we do talk about real-time PCR, that's detecting products during the reaction instead of waiting until the end to see the result. So you can know whether or not it is going to be positive during the actual reaction themselves. We can also use it to analyze RNA. It doesn't have to only be DNA. Okay, recombinant technology, imitating nature. Using recombinant DNA technology, forming genetic material from one organism and combine it to that of a different organism. It's kind of just what I was telling you guys about. That's genetic, forming genetic clones, cloning my insulin gene and putting it into E. coli, right? That's recombinant DNA technology. An example that they're giving here is genetically modified fish. We have zebrafish that they put genes for GFP, which is green fluorescent protein, yellow fluorescent protein, red fluorescent protein, into their genomes. They can actually use this, not just stupid reasons, they can actually use this for monitoring environmental quality of like water that these are living in, because if it's crappy environmental quality, they're not going to produce the color as brightly. So it's not just a useless, crappy tool. Okay. Recombinant DNA technology, cloning. We will remove a selected gene from an animal, plant, or microorganism, the genetic donor, propagate it in a different host organism, usually inserting it into a vector in order to do that. Now, when I say that, I was talking about taking like a human insulin gene. So we'll just depict that as this here. So insulin gene. And then we can pop that sucker into a plasmid, which is a little bit of circular DNA that carries the genes that can help bacteria become more virulent. But we're hijacking that system. We're not making them more virulent. We're taking the genes out that they would have that would have made us sicker, right? More virulent. Taking those out, instead putting in genes that will code for like human insulin. So that vector is the plasmid. And then the genetic donor would have been people. And that is the gene that we have gotten from the genetic donor, is the insulin gene. Okay. And then we'll put that into a cloning host, which often, like I was saying with this example, we have a plasmid here that we put the insulin gene into. We will put that plasmid that we've engineered into a competent cloning host, which we usually use like E. coli. We can use yeast too, but that's the idea. Okay. We can generate these things called genomic libraries. It's literally the entire genome, the cloned DNA that represents entire genomes of whole organisms that they have available. That is useful because then we can use that to for studying, or for we don't have to wait to clone things up. We already have copies of them available. We don't have to rediscover it or something like that. So let's get into this.
The first step in cloning a gene is to isolate the DNA from the organism that contains the desired gene. The isolated DNA is purified and then fragmented with a restriction enzyme. A restriction enzyme produces staggered cuts in specific sequences in the DNA, generating fragments with sticky ends. Each fragment has a single-stranded sequence of nucleotides on its ends that is capable of hybridizing with DNA that has been fragmented with the same restriction enzyme. The DNA fragments are then incorporated into bacterial plasmids. The plasmid used for cloning has a single restriction site, and when cleaved by the restriction enzyme, generates the same cohesive ends that are in the fragments of the DNA to be cloned. The cohesive ends of the plasmid and DNA fragments now line up, and the enzyme DNA ligase is used to form covalent bonds. The next step is to incorporate the plasmids into bacterial host. The cells can now be grown on a solid medium, and those containing the desired cloned gene can then be isolated.
Okay, that's how that would work. There are steps to make it more specific as you go along, but it just depends on where you're starting from. So common vectors, this is a plasmid. This is a common depiction, a map, if you will, of a common plasmid, okay? And what we have going on here, we have an ampicillin resistance. So this amp means ampicillin. What does that sound like to you guys? That's ampicillin. It's like an antibiotic, right? So that is going to convey ampicillin resistance. Why do we care? Because if we put this plasmid successfully into our E. coli, we can grow it up on media that we added ampicillin to. And if it has this plasmid in it, it can grow. If it didn't take the plasmid up, it should be killed by ampicillin in the media. So that helps us determine if the plasmid got taken up correctly, okay? And then we have an origin of replication that can sometimes be controlled by things like the of certain things into the media on, okay? And then we have an area in here called the multiple cloning site. And it is just what it looks like. Remember how we were talking about ECO R1 and we talked about Hind 3 down here. These are all restriction. Okay. Sites where restriction endonucleases, which are listed here, can cut at. So there's a whole slew of sites that you can cut with these specific restriction endonucleases. So if what you are designing with whatever gene you're interested in, like insulin or whatever, and you pop on the ends like, well, we put SAC1, we will cut with SAC1 like this, and you know that this plasmid has a SAC1 site and you cut it with SAC1, that it would insert right there. So you know exactly where it's going to insert and how it's going to insert and everything. So it's controlled. You know exactly what to predict from it. It's not going to be any surprises at all. This is going to allow you several choices to work with. These are all engineered. We have made them this way. This is not a natural thing. Completely designed. Okay. This is the idea. We have our gene. We've cut it. It's got its sticky little ends on here. We took our plasmid. We've cut it. It's got its sticky little ends on here. We can match those suckers up. The sticky ends from the plasmid match up the sticky ends from the gene. Like that, whatever the other one was, sorry, on the previous thing, SAC1. Okay, just like that. Then we ligate it with the DNA ligase, seals it all back up, makes it usable. And then we treat the E. coli in such a way as to make it want to take up this DNA. Now that can be shocking it with hot and cold and stuff like that. Or adding certain chemicals. There's a lot of stuff we can do to make that cell take up that plasmid. Cells that are apt to take up these plasmids, we call those competent. So you want to add it to competent cells. Okay, so now we've got that plasmid shoved into that competent cell. It has the donor gene in it, like insulin, right? It's going to go through transcription, which is making mRNA. Then ribosomes in that bacterial cell, the E. coli cell, are going to translate it. And no matter what, it should be translating it into the protein of interest because we've engineered it to be so. So if that's human insulin, it's going to make human insulin. And then it's going to make that all up in its little media that it's growing up in. And we can purify that out from the flask that it's growing up in. So that's how we would get all that. Don't worry about how we purify it out. That's a whole other class. So we would use this technology making recombinant organisms like that E. coli that we just put that insulin into, that is a recombinant organism now. Okay, we can use it for sources of protein products, making more protein or certain proteins produced by certain things. We can isolate specific nucleotide sequences using medications that cannot be manufactured by other means, forcing other organisms to do it. And large-scale manufacture of hormones and enzymes, like we just said, insulin, that is a real thing. The first recombinant product approved for human use was human insulin made by E. coli. Okay, we can also use this for things like interferons. Interferons are peptides, which are like short proteins that are used to treat things like cancer, multiple sclerosis, viral infections, hepatitis, and genital warts. We have interleukins, a similar type of a thing. Little peptides, they are called cytokines. We will talk about this, don't worry, in Unit 3, okay? Don't worry, you'll know what an interleukin and an interferon is, sadly. So, I mean, I know you guys probably don't want to learn it, is why I'm saying it that way. Yes, but these are things that regulate immune function. That means our white cells, like our lymphocytes and things like that, and we use them for cancer treatment as well. And we have things that can control the tumor necrosis factor or make a tumor necrosis factor itself that we can use to treat cancer. We talk about Remicade or Humira. These are biologics, which are, we made, human proteins, and we made them recombinantly. That sounds weird, but that's what I mean. Yes, we made them using recombinant technology so that we can treat autoimmune and autoinflammatory disorders like rheumatoid arthritis, etc. So that's what they're useful for. Here's some examples. I won't go into the details of these. I listed some of these, but if you're interested in looking over this, you are more than welcome to. They'll be in your notes. When we talk about GMOs, we are talking about genetically modified organism. Any organism that has a foreign gene put into it is a transgenic organism. We call them genetically modified organisms. Okay, they have foreign genes put in them. We use recombinant microbes for things like bioremediation. We have altered bacteria so that they can help clean up and break down oil spills and things like that to clean up our environment. It is useful. It is something that has helped tremendously with those sorts of problems. We can also use them for biofuels, drugs with new properties, because we can control our the DNA that we're putting into these organisms. Industrial chemicals, I mean, in a more environmentally friendly manner. I mean, using microbes and controlling the DNA and the genes that we're putting into these microbes is extremely useful in industry, because we can change things down to the exact molecular level. Okay, transgenic animals. We have animal models that are used to study human genetic diseases. If you want to understand a deadly or potentially a very harmful human genetic disease, and it's potentially rare, or we don't have a lot of people that are going to be, useful candidates for treatments with this because they're already dying from their genetic disease. Unfortunately, the best, option then would be using animal models. So we have developed animal models to study certain human genetic diseases. It's just the way that the world has worked. It's unfortunate for the animals, but if we want to save human lives, so this is the price that we might be willing to pay. Now, aside from that, these are systems that we can use to test. They're natural systems that are organic, that have, moving parts, often like ours, that we can use to test these therapies before we're using them on humans who are already dying and sick. We often use sheep and goats to become factories of certain proteins and things we might want to study about people, making the sheep and the goats produce the proteins or whatever. So we can have them excrete human proteins in their milk or their semen, and then use those proteins that a biological organism has made, an actual mammal has made, that we can equate to a human equivalent. This process is called farming. I don't want to know about it, if I'm being completely honest. I just rather don't tell me about these things. Okay. Synthetic biology, creating organisms from scratch, literally. We understand genes and how they can make certain, so if I tell you that AUG in RNA codes for methionine. And I can tell you that, and I can tell you all of the other amino acid sequences. I can tell you all the amino acid sequence, and you might say, well, I have noticed that, when I want to make such and such protein, that it needs this amino acid sequence. Well, I can reverse engineer it so that, now I have the gene for that. And oh, look at that. I have all these genes that I can do that with. You can make entire organisms from scratch using that information. Yes, you can. So people have made artificial polioviruses, artificial influenza viruses this way. And this guy created a self-replicating bacterium using our normal 4 nucleotides, ATCG of DNA, just from scratch, a whole brand new bacterium. Just freaking from scratch. Straight up from chemicals. He had DNA sequences, splattered them out, made whatever wanted his sequences to say, and now a bacteria came out of it. He was able to create life that way, given the right chemicals available. But you can't just make it out of nowhere. But yeah, he has had the DNA to start with, and gave it the ingredients, and then there you go. Now you've got a new bacteria. That's so nuts. So anyways, yeah, it's scary as well. I agree with you. I know you're saying it. So for gene therapy, moving on, certain diseases. Oh, she's going to play with her toy right now, isn't she? You're cute. I am not talking bad about you. I promise. Sorry, she's heard it. Okay. Gene therapy. If you have something wrong with your genes, like you can't produce insulin, you got something wrong with your gene that it's messed up or whatever. The idea here is that I can introduce a new form of that gene that I can put in other cells in your body, and those cells will produce the insulin. That's the idea with gene therapy, right? So whatever. So recombinant, right? We already talked about recombinant human insulin, where that came from, but now we're talking about gene therapy, moving on to the idea of actually incorporating it into your body so that your body makes it. Okay. So repairing or correcting a faulty gene in humans that are suffering from a fatal or debilitating disease to replace that faulty gene. So we can give that person an actual real life, true to honest to God, permanent cure to that dysfunction. Never need treatment again. Let's see. I feel like this is not the video that, okay, well, it is, but this is some sort of ad. Get away.
For sufferers of hereditary blindness, there may be a light at the end of the tunnel. 2 experimental trials have shown that gene therapy could one day cure a rare form of blindness that affects about 2,000 Americans. The research teams tested the treatment on a total of 6 patients. Here you can see how they performed the eye surgery. Repair genes were injected into the eye of this young visually impaired patient. The eye was infused with air, then reinfused with fluid near the end of the surgery. To measure the patient's performance before and after the treatment, he was guided through an obstacle course. Here, you can see how he navigated through the maze prior to the surgery. He bumped into obstacles 6 times and was disoriented twice. It took him 77 seconds to finish the course. 6 months after the treatment, the patient showed a dramatic improvement. He was able to make his way through the maze in 14 seconds without bumping into anything or getting disoriented. He was one of 4 patients that showed an improvement. The treatment also proved to be safe, which has not been the case with previous gene therapy trials.
That's sort of not that comforting. This is not previously the case. To be safe. Anyways, that's the idea, right? So this is a complicated thing, controlling genes and how your body's going to respond to them. It's a difficult thing to approach, and introducing those into the body in a way that's actually useful without causing harm, that's the even more difficult part. So, but this is the concept behind it, and it is actually a useful concept. So how would we do this? We would have a normal gene from a healthy person that we would isolate. We would clone it like using PCR, right? Amplifying the copies, put them into a retrovirus vector like HIV, hijacking its system and its ability to insert its genome into the host, right? Then take a bone marrow sample from the patient who isn't doing so hot. Put the bone marrow with the genetically altered retrovirus, carries the good gene, and that doesn't have any of the virus stuff in it anymore. It's just got a good gene that it's going to make. Then we would take those cells and put them back into the patient. And then we observe the patient for expression of the normal gene. Now, depending on what you're trying to target, you might want to put it all over systemically, or you might want to treat in localized areas like the eye, like we were seeing in the video. So there are a lot of diseases that are going to result from inappropriate expression of a protein at some point. So we're talking about Alzheimer's, a lot of viral diseases that are technically going to stick around for a while. Cancers could be treated with something like this. And then malfunctioning microRNA stuff. Don't worry about that stuff. We didn't talk about that, but anyways, Mike, I guess we're going to talk about it. I forgot that the slide was in here. Sorry. MicroRNAs and siRNA, silencing RNAs. The idea with these guys is they interfere with your mRNA products to make it so that they don't make your protein target. So we can use microRNAs as a way to target specific genes to silence them so that they're not making bad things anymore. So if you have cancer, for example, you might want to silence certain genes so that you can stop cancer protein production. Well, proteins that are related to cancer anyways. So yeah, so that's the idea, silencing them, turning them off. That's what microRNAs and siRNAs are going to do, whatever. So that's the idea with gene therapy, either changing the genes itself or introducing microRNAs to silence the genes. What we've talked about in this chapter, we talked about cloning things. We talked about recombinant DNA, putting a new gene into a vector of some kind, putting that vector into a host, and then having that host produce our target gene product, right? Ligase, remember the ligase, Ligase is going to seal everything up, right? Seal back together those nucleotides so that it can function. PCR, polymerase chain reaction, just copying DNA pieces. A lot of them. We can get a lot of new copies from one source, one very, very small amount, right? Know the steps of it and know what is needed for it. Know the steps of it, know what is needed for it. We have denaturation, usually that's heat, splitting those double strands apart into single strands. Annealing, by annealing, that's bringing back together. And here we're talking specifically about primers. The primers are going to bind to their specific sequences there. And then we're going to extend using that DNA polymerase from those primers starting as a starting point, right? And then we talked about gene therapy and fixing people. So that is that for chapter 10. I hope that wasn't too awful for you guys. We have chapter 24 and 25 as one kind of unit next, and then that's it for unit 2. I'll see you guys next time, I guess. Sounds like fun.